Hello everyone, I welcome you all to the PYQ session of Biotechnica, and today we are going to discuss about questions from Unit 2 from CSIR NET Life Science syllabus. And the topic over here is cell organelles and protein transport. So before proceeding with the question discussion, let me mention you one thing, that protein transport is one of the important topic from examination point of view. So every time, every exam, you will expect at least one or two questions specifically from the transportation part. And that could be in the form of experimental questions, or in the form of match the following, or it can also be in the form of statement based questions. So today I am taking a few organelles. Remaining organelles based questions I'll take up in another session. So the very first question over here is, thousands of proteins are synthesized in the cytoplasm, are imported into the nucleus through NPC every minute, that is nuclear pore complex. These proteins contains NLS that direct their selective transport into the nucleus. This nuclear import requires a small monomeric G protein RAN, a nuclear transport receptor that interacts with NLS on a cargo protein, a GTPase activating protein bound to the chromatin tethered to the nuclear membrane, and the last statement is a guanine exchange factor bound to the chromatin inside the nucleus. Now, we need to identify the correct combination of statements out of this. So, the options will be provided to you, and here as you can see, the correct answer is statements A, B and D. So, let's check it out. What are the statements A, B and D are? So, for a nuclear import to take place, definitely we require a RAN protein. And RAN by nature is a monomeric G protein. Second, the nuclear transport receptor interacts with NLS on a cargo protein. NLS serves as a sorting signal for a protein to enter inside the nucleus. So, in order to carry the cargo inside the nucleus, you must have a nuclear transport receptor. And that receptor name is, or I can say they belong to a family of proteins. We call it as a karyoferrin. And this karyoferrin is again divided into two parts, import receptor and export receptor. That means import receptor will carry the cargo inside the nucleus. Export receptor will carry the cargo outside the nucleus. That means towards the cytoplasm. Then we have option or statement D as correct answer. That is, Jeff is involved in the transportation which is bound to the chromatin. So obviously when you look into the statements over here, C and D, one of them has to be true. So if Jeff is present in the chromatin, then what about gap? Where it is located? Let's have a look into that. See. This is the image that explains about the RAN protein. As I told initially, RAN is a very important protein required for import and export of the cargo molecules towards the nucleus or outside the nucleus. So this RAN protein exists in two forms. One is the GTP bound form and another is the GDP bound form. So GTP bound form is considered to be the active form and GDP bound form is considered to be the inactive form. So in what situation RAN is bound to GTP and in what situation RAN is bound to GDP? Let's have a look onto that. So as you can see here, we have cytosol and this is nucleus. So within the nucleus we have chromatin present. So in this chromatin, you will find RAN-Jeff is located. RAN-Jeff means RAN guanine nucleotide exchange factor. So guanine exchange factor means some exchange is taking place. What is the exchange? GDP is going out and GTP is coming in. So what we are getting? We are getting RAN GTP. And RAN GTP is the active form. So remember, RAN GTP is catalyzed by Jeff and the process takes place inside the nucleus. Similarly, on the other hand, we have RAN gap located. Where? Within the cytosol. RAN gap is RAN GTP's activating protein. GTP's activating protein means what? That are involved in GTP hydrolysis. So as we can see, inorganic phosphate is getting released. So what we are getting? RAN GDP. So RAN GDP is considered to be the inactive form catalyzed by gap. And where it is located? It is located in the cytoplasm. This is a very important concept. You must remember either in the form of a flowchart. Flowchart, in fact, I will say flowchart is better to be remembered because that will give you the insight and it will be easy for you to remember the content. So, that's the reason statement C is incorrect, statement D is correct. Moving on to our next question. As I have mentioned before, you can expect questions also in the form of match the following. Now, if you get these kind of questions, definitely it's a scoreable question. The reason is that everything is given directly. This particular question involves cell organelles on column A and their corresponding signal sequences on column B. So you have to identify the correct combination over here. And the correct answer over here is option 3. So as we can see, lysosomes. Always remember one thing. Lysosomes are the only organelles that does not have amino acid as their sorting signal. Remaining all the organelles, or I can say majority of organelles, will have amino acid sequence as a sorting signal. So among these combinations, you see we have mannose-6-phosphate, right? So lysosomes to mannose-6-phosphate, A to 4. So you got one combination which you are confident on. Next, mitochondria. Mitochondria and nucleus. If you take into consideration what is the similarity between them, they have got amino acid sequence that is common. For example, lysine and arginine residue, here also lysine and arginine residue. But what is the difference is that, remember, the conserved amino acid sequence for mitochondria will be located towards the N-terminal region of the protein. Whereas when it comes to nucleus, it is quite unpredictable. It could be N, it could be C, it could be anything. So that's why you can easily differentiate among these two. Next, what we are having? Peroxisome. So the leftover is peroxisome. Peroxisome will be having two signaling sequences. Here also you have the N-terminal signaling sequence. But what is the amino acid? That is not given. They have just mentioned it as a 9 amino acid sequence present towards the N-terminus. Otherwise, the protein or the polypeptide should have a C-terminal tripeptide sequence. What is the tripeptide? We call it as an SKL sequence. Serine, lysine, leucine. It is present towards the C-terminus. So either of this signaling sequence can serve as a sorting signal for a protein to enter inside the peroxisome. Remember, this is otherwise known as PTS1, that is peroxisomal targeting sequence 1, and this sequence we call it as PTS2, peroxisomal targeting sequence 2. So we have a table regarding that. As you can see, target organelles, their sequence in which location they are having, and whether the sequence is removable or not. If you see, in case of peroxisome and nucleus, the target sequence or the sorting sequence or the sorting signal is not removed. It will be retained along with the proteins. But remaining organelles, if you check up, endoplasmic reticulum, mitochondrion and chloroplast, the signaling sequences are removed once they reach the destination. That means they will be having the specific proteases or peptidases that will help in the degradation. And all the proteins will have N-terminus. That means the signaling sequence will be present towards the N-terminus. So, if you see the endoplasmic reticulum, it is a core of 6 to 12 hydrophobic amino acid preceded by one or more basic amino acid. For mitochondrion, it is an amphipathic helix, 20 to 50 residues in length, that means quite long. That will have arginine and lysine on one side, whereas hydrophobic residues on the other. So here hydrophobic amino acids preceded by basic amino acids in case of endoplasmic reticulum. But in this case, two ends, one end will have the basic amino acid and the other end will be hydrophobic amino acid. When you look into the chloroplast, they are also N-terminus. They do not have any specific motif, but it is believed that they are rich in serine, threonine and small hydrophobic residues, but poor in glutamate and aspartate. So, as I had mentioned previously, peroxisome, PTS1 and PTS2 signal. If PTS1, it will be towards the NC terminus. And if it is PTS2, it will be towards the N terminus. Next, the next question. Which one of the following proteins is most likely to be found in the intermembrane space of mitochondria? We are aware that mitochondria is having 4 compartments. That is outer mitochondrial membrane, inner mitochondrial membrane, then we have intermembrane space, and then we have the mitochondrial matrix. So in all the compartments, the proteins must enter inside. But here what they are asking, you have to identify the intermembrane space of mitochondria. That means the proteins that should enter into the intermembrane space. A N-terminal matrix targeting sequence followed by hydrophobic stop transfer sequence. An N-terminal matrix targeting sequence followed by cleavable hydrophobic sequence that blocks the complete translocation. A protein with multiple internal sequences being recognized by TIM22 complex. A protein with an outer membrane localization sequence followed by a matrix targeting sequence. So you need to identify the protein which is entering into the intermembrane space. So the correct answer over here is statement 2. That means an N-terminal matrix targeting sequence followed by cleavable hydrophobic sequence that will block the complete translocation. Now why statement B or statement 2 is correct? Let's have a look onto that. For this, you need to remember the arrangement of targeting sequences present within the mitochondria. So if you see, if a protein needs to enter inside the mitochondrial matrix, they should have a matured protein with an MTS. MTS means matrix targeting sequence. Then only it will enter inside the matrix. If we compare it with the proteins from inner mitochondrial membrane, we have 3 different pathways for that. So we have 3 different types of sequences. What are they? The very first type of polypeptide is, it's a mature protein with hydrophobic stop transfer sequence and an MTS. That is matrix targeting sequence. So once they reach the destination, it will be cleaved. So this stop transfer sequence will help in transferring or anchoring it to the inner mitochondrial membrane. Second kind of protein that is directed towards the inner membrane is, again, they will be having the matrix targeting sequence. Then they have some internal sequences which are being recognized by a group of proteins. We call it as an OXA1. OXA1 is basically a translocator which is present in the inner mitochondrial membrane, and they will recognize any protein if by mistakenly enters into the matrix can be retrieved back to the inner membrane. And the third pathway is all about a polypeptide that does not have MTS. That means they do not have any exposure to the matrix. They have some internal sequences that will be recognized by TOM70 as a receptor and TIM22 complex. That means the general import pore and the import receptors present in both outer mitochondrial membrane and inner mitochondrial membrane. Then we have intermembrane space targeting proteins. So this is also of two types. As you can see, we have two polypeptides over here. So in the first case, you will have two types of cleavage. One is MTS cleavage, that means they have exposure to the matrix, so the MTS will be cleaved. Matrix targeting sequence will be cleaved. But in the second case, you will find intermembrane space targeting sequences there. So that will be the second cleavage. Second type of polypeptide that enters into the intermembrane spaces, they do not have MTS. So what happens is that the targeting sequences directly enter through the general import pore. So they are not dependent upon the outer mitochondrial import receptor or inner import receptor. They are directly entering inside the intermembrane space via general import pore. Then if you look into the outer membrane proteins like porins and all, they do not have any specific MTS, but yes, they will have a stop transfer outer membrane localization sequence. So that will help the polypeptide to be tethered to the outer membrane. So the answer over here was an N-terminal matrix targeting sequence followed by cleavable hydrophobic sequence that blocks the complete translocation. So that is this one, this polypeptide, right? So it is important to remember the arrangement of polypeptide chain, specifically the cleavage patterns when it comes to mitochondria. The next question we are having is again another match the following kind of question. As you can see, you have cell organelles on one side and some vesicles or transportation on the column B. So as I mentioned before, lysosomes is the only cell organelle that will have mannose-6-phosphate. So corresponding to that mannose-6-phosphate receptor will be present. Next we are having cis-Golgi, trans-Golgi. So cis-Golgi, you will find that from there the vesicles that originate is COP1 vesicles. And trans-Golgi is a Golgi from where COP2 vesicles are originating. Remember this part. Okay. So we have cis-Golgi, COP1 vesicle binding. Trans-Golgi from where COP2 vesicles are originating, but since that is the last part of Golgi, usually the proteins are aggregated for secreting out to different organelles or maybe fusing with the membrane. Then COP2 vesicles, they are involved in anterograde transport from ER to Golgi. So always remember we have two types of transport mechanisms, anterograde, then we have retrograde. So anterograde is one that will be moving in the forward direction. That means from the ER it will move towards the Golgi. Whereas retrograde is the one that moves in the reverse direction. That means from the Golgi back to the ER. So if you remember this concept, things are better. So COP2 vesicles are involved in anterograde transport from ER to Golgi. So you can remember this is COP2, right? And you can remember this is COP1. So COP1 is involved in reverse direction, COP2 in the forward direction. Then we have endocytic vesicles, that is we can connect it with clathrin coated vesicles. Right, clathrin coated vesicles have origin either from the plasma membrane or from the trans-Golgi network. So usually they are involved in receptor mediated endocytosis. So for this also we have a table. See, COP2 vesicles, as I told you, moves in forward direction, and these are some coat proteins or accessory proteins that helps in the transportation. Along with that, we have a specific GTPases associated with them, that is SAR1. But for COF1 and clathrin coated pits, remember we have ARF serving as the GTPases. So COF1, Golgi to ER, that means in the reverse orientation. So this is anterograde, this is retrograde. So here you will find some coatomers are present that serves as the coat proteins. Clathrin and adapted proteins, we have trans-Golgi to endosome catalyzed by clathrin and AP1 complex. Trans-Golgi to endosome can also be catalyzed by clathrin and GGA sequence. And as I told you, it can be also originating from plasma membrane as well. So if it is from plasma membrane to endosome, clathrin plus AP2 complex. So remember the difference. If it is from Golgi compartment, it will be to lysosome, melanosome and all. Then remember it is AP3 complex. So this table is also important from examination point of view. So that's it for today's session. In the next session, we will be having some more insights about the remaining organelles like ER, Golgi, and I'll take up some experimental questions as well. Thank you so much.